Hello, my name is Ailet Vargas, and I'm going to talk about the Kirin amendments eliminate the negative effects of continuous cropping, obstacles on soil properties, and microbial assemblage. As an introduction, we have that the soybean is an important crop in China. Given that soybean is highly susceptible to continuous cropping and increasing frequency of soybean monoculture might result in an excessive soil health de- deterioration and substantial losses crop yield. While previous studies have documented that biotic factors rather than abiotic factors are main reasons for the occurrence to continuous cropping obstacles of soybean. And the traditional control practice for continuous cropping obstacles including the use of crop rotation and chemical agents still have potential drawbacks. Crop the rotations requires a long periods to reduce soil-borne pathogens and eliminate the negative effects of the continuous cropping, and its effectiveness and it's limited once the disease breaks up. And the chemical fumigants and fungicide have high efficiency in killing the soil microorganisms, but the intensive use of chemicals not used cause severe environment pollution, but also encourage the development of target pathological resistance. Now we have the chitin. The chitin is the most abundant polysaccharide in the nature. It possesses good biocompatibility, biodegradability, and non-toxicity and various biological activities and poses no hazard to the environment. Um, one more importantly, the kitten also has a great potential for improvement soil quality, inducing plant disease resistance and promoted the plant growth. And the kitten have been reported to promote the plant growth. Now we have the material and the methods. This study was established on May 10th of 2019 at the Experimental Garden of Northeast Institute of Geography and Agroecology in Chinese Academy of Science. This was in China. The exogenous kitten of pure chitin and crude chitin were purchased from Gianapolo Cruz Tension Fertilizer and Shanghai Hike Xingwu Biotech respectively, and they were used to assess the effectivity of exogenous curing on continuous cropping obstacles. There were seven treatments with different curing addiction ratios were designed in this study. The addiction ratios of purkitin were of 0.05%, 0.1%, and 0.2%, while of those of crude creatine were 0.5%, 1%, and 2%, which were calculated based on the soil mix ratios used in each boat. And these treatments were marked as PC1, PC2, PC3, CC1, CC2, and CC3 of purkitin and crude ketine respectively. Well, were in total it was of 56 bulks and 56 responsive soul samples were collected. Is 7 treatment per 8 replicates. And a single bulk soil sample was recollected from each pot and sieved through 2 mm mesh to fully homogenize and remove the impurities. Book soils were placed into sterilized 50 mm centrifuge tube, and the remaining soil was put in a reusable bag. The reusable soil samples was collected by shaking methods to perceive the soil firmity attached the root surface, and a portion of each collected resource per soil sample was placed and sterilized at 2 millimeters centrifuge tube, and the centrifuge tubes were kept at minus 80 degrees Celsius for subsequent molecular analysis, while the rest of the soils and soybeans plants were temporarily kept in a 4 degrees refrigerator prior to downstream determination for the determination of the physicochemical properties and soybean treatments morphological characteristics. Now we have the measurement and the analysis of chemical properties. We'll consider the quantity of the collected resource per soils was too small to determine the chemical property. Soil chemical properties were measured only on weak soils. Soil pH was determined by pH meter. The soil total carbon and nitrogen were assayed by the combustion method using a CNS elemental analyzer after dried soils passed through the 0.25 millimeter sieves. After the soil samples were measured by sodium hydroxide, the soil total of phosphorus and potassium content were determined by the molybdenum, blue metal, and flame spectrometry method. Respectively, the ammonium and nitrate of nitrogen were extracted, and the ammonium nitrogen and nitrate of nitrogen contents were analyzed from the supernet using a continuous flow analytical system. The, the soil available of phosphorus and potassium contents were quantified with molybdenum, blue, and flame photometric methods. The soybean plant morphological characteristic and soil total microbial activity measurements. Well, the plant height was determined with a tape line to measure the length from the soybean cotyledon scar to the top growth point of the main stem. Well, they cut the plants from the cotyledon scar and and weighted and weight sorry then to obtain the plant fresh weight. The root length, root superficie, and the root superficie area and root volume were measured using a root analyzer. After root noodles were completely removed with tweezers, the fresh soybean plants were and roots were unwrapped into envelopes desiccant at 105 degrees for 30 minutes. Switched to the 80 degrees for 40, 48 hours, and finally waited to measure the plant and the root dried weight. And the total microbial activity of the wet soil was determined by the fluorescein diazotide hydrolysis method. And first, they have the 15 millimeters of the potassium phosphate buffer and zero point 0.2 millimeters of FDA stock solution. They were added to 2.0 grams of result to initiate the hydrolysis reaction. Then, after the incubation at 30 degrees for 20 minutes, and 50 millimeters of chloroform methanol was added to terminate the hydrolysis reaction. Finally, the absorbance of the supernatant was determined with a spectrophometer with a wavelength of 490 nanometers. and the fluorescein content was calculated according to the standard curve to obtain the full total microbial activity. The DNA extraction, PCR amplification, and hydro well, the soil microbial DNA from each sample was extracted for zero point 0.5 grams of fresh soil using the fast DNA spin kit for soil. According to manufacturer's manual, the extracted DNA was measured for concentration and purity with a nanodrop to 2000 spectrophotometer, and the quality of extracted DNA was examined using 1% agarose health electrophoresis. And the PCR amplification was conducted in a 25 microliters reaction system comprising 2.5 microliters of DNTPS and fast PFU buffer with zero point 0.2 microliters of BSA and zero point 0.5 microliters 5PF polymerase and zero point 0.5 microliters forward and reserve the primers and templates the DNA and sterilize the ultra-pure water. Well, each sample was amplified and triplicated, and the PCR products were combined and purified using agarose gel DNA purification kit, examined in a 2% agarose gel, and quantified by a quantifluor photometer. And this was following the manufacturer's protocols. The sequencing data analysis, the raw data from the sequencing were analyzed using the KIM QIIME 1.9.1 and the merged through Flash 1.2.7 software and considered the impact of the sequence number of variation among the samples based on the minimum sequence subset of 18 18,302 and 29,755 sequences from each sample of bacteria fungi were randomly selected for sequence analysis. And the structural equation modeling was constructed to assess the direct and indirect influence of the prominent soil chemical properties on beneficial microbial taxa, potential pathogens, the fluorescein diacetate, and the plant growth performance. The structural equation modeling were carried out using the IBM SPSS Amos 21.0 software. Now we have the results and the table number one. That this is one from the soil chemical properties under different of the pure and crude Chitin addition to such into bulk soils. Well, we have the treatments with the pure Chitin and the crude Chitin, and we have the rate of the 0%, 0.05%, 0.1%, 0.2%, and 0.5%, 1%, 2% of that soil mass respectively. And now, pues, the Kirin amendments significantly improved that the soil chemical properties compared to that in the control. Well, the crude Kirin remarkably elevated in the soil of the total nitrogen in the crude Kirin 2B and the crude Kirin 3B, but the soil to total carbon and CNA radio content were significantly reduced by the pure Chitin and the Chitin amendments, with the exception of the total of the TC in the crude Chitin 3B. In addition, the Chitin amendments sharply decreased the soil nitrogen, while it was highly increased in crude Chitin 2B and crude Chitin 3. And compared with the COB soybean plant morphological characteristics such as the plant height, root dry weight, and the absorbent ground dry and fresh weight were significantly increased under the pure Chitin and the crude Chitin amendments. In addition, with the exceptions of the pure Chitin 2B and the pure Chitin 3B, the soybean root volume was significantly increased in the both of the pure Chitin and the crude Chitin amendments. However, there were no significant difference in such plant morphological characteristics amongst the difference of the pure Chitin addition dosage. Now we have the figure 1, and this figure is the non-metric multidimensional scaling plot of all soil bacterial and fungal communities. The separate NMDS plots shows the effects of the pure and crude Chitin amendments on the bacterial and the fungal community structures in the bulk of the rhizosphere soils, respectively. We have the CO in the PC1, PC2, PC3, and the CC1, CC2, CC3 represents that the pure and the crude Chitin amendments rate at the 0%, 0.05%, 0.1%, 0.2%, and 0.5%, 1%, 2% of total soil mass respectively. B is for bulk soil and R for the resource per soil. And the better diversity of microbial community illustrated by NMDS plots clearly shows that the bacterial and the fungal communities were obviously divided into two major groups, that is, different kinds of Chitin amendments, whereas the microbial communities in the bulk and the resource per soils were grouped closely. In addition, the separate NMDS plot shows a clear separation between the different dosages of pure and crude Chitin additions, which also differ from the control. Well, we have the second image that, well, this represents the change in the dominant bacterial and the fungal taxa. In total, 6 million good quality bacterial sequence and 7 million good quality fungal sequence were obtained from the 112 soil samples in the study. When the group at the 97% similarity threshold, these sequences were clustered into 9,113 and at 2,611 OTU for bacteria and fungi, uh, which were identified by to 36 and 8 phyla respectively. The bacterial and the fungal community compositions in the bulk and the results per souls under the different pure Chitin and crude Chitin amendments were shown in this image. Well, this image is the relative abundance of the dominant bacterial and fungal phyla class, and it was at least one treatment, less than 1%. Now we have the figure number 3, and this one is for the relative abundance of potentially beneficial bacteria under the different pure and crude Chitin addition dosage. The bars indicates the means of the eight replicates, and the error bars represent the standard deviation. And the different letters indicate the significant of difference among treatments. Now we have these graphics, and this is the figure number 4, and the letter A and the letter C represents the relative abundance of the potentially pathogenic, and the letter B and letter D the beneficial. The fungi under different pure and crude Kirin addition dosage. The bars indicate the means of the eight replicates. The error bars represent the standard deviation, and the different letters indicate the significant difference among the treatments. Now we have the image, the figure number 5, and this represents the subnetworks of the potential plant pathogens separated from the microbiological network under the different pure and crude Kirin additions dosage in the bulk and soils. And the circles and the diamonds represents the bacteria and the fungi, and respectively the size of each node is proportional to the degree, and node with blue, orange, and gray indicate the probiotic, pathogen, and other. And the microbial taxa are labeled in the blue and orange nodes. The blue and the red lines show us the positive and negative interactions between the two individual nodes respectively. And this is the figure number 6. This is similar to the figure number 5, only that this is different because is in the rhizosphere soils. And same, the circles on the diamond represents the bacteria and the fungi respectively, and the size of each node is proportional to the degree. Node with blue, orange, and gray indicate a probiotic, pathogen, and other respectively. The microbial taxa label with blue, orange, and nudes, and the blue and the red lines indicates the positive and the negative interactions between the two individual nudes respectively. And to conclude this presentation, we have that the authors of this article investigating the impacts of the Kirin amendments on soil properties, microbial communities, and soybean growth in a soil with continuous cropping soybean. And they found out that the pure Kirin and the crude Kirin are greatly increase the soil pH and available nutrient content, and improve the soybean plant growth and soil microbial activities. The Kirin amendments remarkably enrich the potentially beneficial bacterial, fungal, and taxa reduces the potential plant pathogens. And this implies that the Kirin additions may contribute the, to the development of the healthy soil microbiota. In addition, the Kirin applications cause a looser pathogenic subnetwork structure, which demonstrated that the Kirin amendments may mitigate the spread of ecological risk and potential plant pathogens in soil systems.